Thanks again. The next speaker of the session is Dr. David Muddiman. Dave is a Jacob and Betty Belin Distinguished Professor of Chemistry and the Director of the Molecular Education, Technology, and Research Innovation Center at North Carolina State University. Today, he will present on quantifying human exposure to emerging contaminants of concern using mass spectrometry.
Okay, thank you, Andrew, very much. And I really appreciate the invitation today. First, I want to thank my graduate students and collaborators over the years, funding from mostly from NIH and NSF. We have a lot of NC State and academic collaborators, but in this particular case, a lot of what we do, we could never do without the help and collaboration, collaborative spirit of CIL. So we really do appreciate that. So in terms of environmental exposure, so it's an area that we spend a lot of time on at NC State University with the Center for Human Health and the Environment. And we talk about life basically interacts with genetics to give a response surface. So like we say, life is lifestyle, infection, food, or exposure, interacting with, as we learned from the trivia, 99.9% common DNA. And then you can look at different response surfaces, such as the genome, epigenetics, for example, metabolomics, proteomics, and so forth. So we look at lots of different environmental exposures, trying to understand diseases that just don't seem to have a genetic link. And then we try to bring these data sets together, collected from other people like genomics and transcriptomics, which we don't do, but then we do the other omics and try to pull these data sets together, which is much more difficult than it might seem. So the part one of this talk is the role of the environment in ALS. And so if you look back over the past 38 years in people studying the role of the environment in ALS, there's only been a 3-fold increase in the number of publications during that 38-year time period. But when you look at genetic factors with the rapid sequencing and whole genome sequencing capabilities, there's been a 71-fold increase in 24 years. Now those papers have a lot of suggestive genes that might play a role, but there's been no conclusive new information that's really come out except for the main 4 genetic mutations the 4 genes that actually are linked to ALS. So ALS is a rare disease. It's a diagnosis by exclusion. So one of the things that happens is it takes about 14 months from the time you have symptoms to where you finally get an accurate diagnosis that you have ALS. And so we feel like that window is the window of opportunity for even experimental treatments. And by the time you've been diagnosed, the progression of the disease, since the median lifespan is about 3 to 4 years, that you've already missed that window of opportunity to try to reverse the effects of motor neurons dying. So we're trying to find new, faster diagnostic treatments, but also try to understand the origin of the disease at the same time. So when there's been hypotheses that have been mulling around for 40 years now, dating back to the study in Guam, and these are just a collection of papers that all use non-specific methods for detecting a neurotoxin that's produced by cyanobacteria, and that's called BMAA. All of these methods use that ELISA test, and we have now documented within our own lab and tests and so forth that this gives extraordinarily high levels of BMAA even when it's not even in the sample. And so there's a lot of false positives out there. And so these series of papers that have been published, these are just a handful of them, there's a lot more, really suggest that BMAA could be a player because of these cyanobacteria that grow in fresh bodies of water. So here's a case off the coast of South Florida. It's a blue-green algal bloom. It's quite extensive. In a paper that was published, they published that it was 1.5 milligrams of BMAA per gram of shrimp. So it's a very plausible route of exposure because now you have cyanobacterial blooms. Shrimp are in the water, you eat the shrimp, and now you have a transmission to the human population. So it all sounds very plausible, but again, these are non-specific methods. In Lake Erie, this is a 92-kilometer wide cyanobacterial bloom. And this actually happened back in the spring of 2014 because what happens is all the over-fertilization of land, when there's significant amounts of rain, that washes into these bodies of water, which already have cyanobacteria, but this just makes them grow and thrive tremendously. And some cyanobacteria do, it is well known that some cyanobacteria do produce BMAA as well as many other neurotoxins. So we formed a team of people from synthetic chemistry with Josh Pearson in my department with his graduate student at the time, Yasmin. And then my group members, Kaylee and Phil, working with Michael Behrman and his graduate student, Josh, and an expert in harmful algal blooms, Astrid. And so we wanted to set out to begin with to make a specific internal standard. And so we worked out the chemistry in Josh's lab with him to actually make a stable isotope labeled version of BMAA. And so this just shows you the molecule that we made. When we do tandem mass spectrometry on it, we know that all the labels are where they're supposed to be. I will tell you, and it's not against Josh Pierce. He's not an amino acid chemist, and so I went to CIL and I asked them, "Can you make this for us?" And because I think the field really needs it to rule in or rule out this hypothesis, because the material that Josh made was chemically and isotopically not so pure. So CIL made the molecule. You can buy it today, and I urge people when they're doing studies with BMAA to use this standard, because at least we can try to get reproducible results and find out what's really going on inside of bacterial blooms. So we have the internal standard. We can rapidly separate it from different isomers of BMAA using the zip chip. So this is capillary electrophoresis in about just a little over 1 second, 1 minute measurement. So it's a very rapid measurement. It's quantitative, and here you can see the unlabeled material in the green, and then the labeled stable isotope label in the purple highlighted box. And so we can actually then just do absolute quantification. So we published that paper and back in 2018, and a group in Australia, which was connected, I got connected to through a friend of mine named Mark Baker. So Gilles contacted me and said, "Oh, can you run our samples? Because in these bodies of water all around Australia, there's a lot of hot spots." And so this is just showing you the deaths per 100,000 people per year, and it's climbing since the late 1950s in males and females. And that's not genetic drift. But BMAA has been detected again with a non-specific method, but they detect these near these ALS hotspots. And so it's higher than the prevalence than the national average. So they sent us this really nice collection of human specimens. There was 252 plasma samples in the experimental design. We had control samples and sporadic ALS. I should have mentioned that genetic links to ALS is less than 10% of the cases. 90% or more are sporadic, no known cause. So we had these controls and sporadic. We had reasonable proportions of males and females. We carried them through a very stringent QC process with positive and negative controls and everything. The data looked fantastic. We did it on the zip chip with a Q-exactive HFX, so RAM mass spectrometry, and of course we use Skyline to do all of our quantitative processing that comes out of the McCoss laboratory in Washington. We got beautiful spectra. We see all kinds of metabolites in these samples, but BMAA wasn't detected in a single one of those samples. And so we tried to look at some of the other data using Compound Discoverer. So in general, we see about 150 metabolites in just a little over 2 minutes. So this is a little bit longer electrophoretic run to get more resolution. But we see about 200 metabolites. We looked at all those different metabolites, and there are other ones on this list that struck our interest in terms of predictability. But one that really stuck out was creatinine and creatine. Now, this is going to be most likely a downstream effect from muscle wasting, from motor neuron death and muscle wasting. But it's interesting because phosphorylated creatine makes creatinine and ATP. And of course, that's an enzyme called creatine kinase, and that's actually an experimental treatment for ALS patients. And so that was actually quite interesting. So this might turn out to be some way to get earlier diagnosis of ALS patients. If you look at the controls versus the sporadic ALS in the males and the females, you can see the p-values in terms of predictability between the controls and the ALS samples are on the order of 10 to the minus 12, 10 to the minus 13. And this isn't the first time this has been seen. We weren't really looking for it. It just stood out in the data set, as well as many other things that we're currently pursuing. So, so that's a part one of the story in terms of BMAA. And again, with that internal standard, we were really allowed to develop a very specific and quantitative method to unravel whether or not we could detect this neurotoxin in the patients of in ALS patients. So the second part of my talk is a different type of exposure, and this has to do with the quantification of perfluorinated alkyl substances. And this work is done with Jeff Enders, a graduate student in my group, Grace O'Neill, and a colleague of mine, Jerry Whitten. So we wanted to develop a new liquid chromatography, electrospray mass spectrometry methodology in support of a new Superfund grant that we were awarded here at NC State. There's over 5,000 PFAS compounds known to date, and these compounds enter into the waterways because companies who make Teflon, they basically, their wastewater goes into there, and then this water just runs, and then people, gets into drinking water. And at wastewater treatment plants, PFAS is not purified out. And so there's a growing accumulation of these. They're persistent molecules. They do not degrade in the environment. And so it's a very important health thing, which has been known, again, for 30 years. It's just trying to quantify these in the state of North Carolina because of a plant that we have here. So the methodology is built on a thermo instrument. It's an Orbitrap Exploris 240. It has that new easy IC internal calibrant source, which gives us high mass measurement accuracy. And of course, then we can fragment things with HCD and get MSMS spectra. So this is dedicated to this project because of the large numbers of samples that we are going to be running this method on. So internal standards are very important, as you know. And one of the things is that I mentioned there's already 5,000 compounds in the environment that are known, but there's no way we're going to develop 5,000 internal standards, stable isotope labeled. I'm sure CIL would love to do that, but that would break the bank. And so what we did was we set up a internal standardization process by which we have matched internal standards, but then we have good surrogates, and we wanted to test that out. Now, if you think about PFAS, and they're very curious molecules, because if you think about small chain fatty acids, like shown on the left here, that these small chain fatty acids are weak acids. They are known to dimerize, and you actually derivatize these if you want to measure small chain fatty acids by LCMS. But the PFAS equivalent is actually a strong acid, has a pK of about 0. And it also has a very large dipole moment. So on the bottom here, what I'm showing you is what the surface of a droplet might look like. So these are ionized in negative ion mode. So through the reduction of water, we have hydroxide anions on the surface. But the PFAS molecules themselves, they're going to repel one another. So they're going to occupy the droplet of that surface at a much lower concentration. And so when does the bulk concentration when does the surface concentration no longer represent the bulk concentration? We think it's going to happen faster for perfluorinated compounds. So just to give you an idea here, looking at on the left-hand side here is just hydrocarbon species. On the right-hand side is the perfluorinated equivalent. And if you look at like log P or nonpolar surface area, you can see just with 12 carbons, and still 12 carbons on the right, but they're perfluorinated compound, the log P value is 400 times. So it's 400 times more hydrophobic. And so that's what really drives the electrospray ionization process, where you're going to stack those droplets with hydrophobic species. And so PFAS are going to have a surface activity close to unity. And so, and where the hydrocarbons are going to be more distributed in the entire droplet. So we wanted to see if we could get away from using nonpolar surface area and log P and just do something very simple and just count the number of fluorines in a molecule. So this is showing you nonpolar surface area versus log P. This is numbers of fluorines versus log P in blue. And this is number of fluorines versus nonpolar surface area. The reason being is because nonpolar surface area, you can calculate this in any number of ways. Log P is not experimentally determined for every molecule, but a prediction based on other databases. And so trying to standardize this and just counting fluorines, which any of us can do, and we will all come up with the same number, would seem like a very logical thing. And it turns out it's all really correlated. And then if we look at log P versus retention time by reverse phase chromatography, you can see that nonpolar surface area in blue and log P in red And then over here on the right-hand side, number of fluorines versus retention time. So everything basically follows the same predictability. So we've now switched to just looking at numbers of fluorines in a molecule, and that's important when you're thinking about surrogate internal standards. So you'll want to match your numbers of fluorines because it's going to change your ionization efficiency for each individual compound. And so when you're running thousands and thousands of environmental samples or human exposure samples, which we are doing, you have to have a relatively rapid method. And so co-eluting isn't really, they're not right on top of each other, but there's a lot of compounds that are coming out in a 10-minute retention time window. And so we really just need one calibration curve where we break it into two. How steep can we make our chromatographic gradient, things like that. And then how do we choose the appropriate surrogate internal standard when we don't have a stable isotope labeled version available? And there's a lot of stuff in the PFAS literature where people talk about method robustness, so these occurrences where molecules, analytes just disappear, and they just have no idea why. So we had to really kind of unpack this entire method and start completely over with what we wanted to do. So this is showing you an example of PFOA with a matched internal standard. And so this is R squared value versus the number of fluorines in the molecule. And so you can see that R squared is basically 1. But as you move forward with less or more fluorines, you can see there's actually a pretty rapid decay. And over here on the right is the calibration curve with its matched internal standard. On the bottom is when it's matched to a different internal standard with a different number of fluorines. And you can see the R squared value goes to negative 2.2. So very poor correlation, very poor quantification will result. So what we did with in collaboration with CIL is we made up 2 Superfund mixtures, 1 and 2. And so on the left-hand side here is the compound name, and then on the right-hand side is a matched stable isotope labeled internal standard. And you also see the number of fluorine counts. And of course, all these in stable isotope or in Superfund mix 1 all match. On the right-hand side are ones that do not have a stable isotope labeled version available at this time. And so on the right-hand side, you can see the number of fluorines, and we try to match that up as closely as we possibly can, because we have found through other studies that that's obviously it's very important in terms of electrospray response. Another great benefit of these compounds is that there's several other companies that sell fluorinated PFAS compounds. One of those is Wellington, and they sell nice products, but the mass shift isn't very large. And so if you look here with the yellow ones, the ones that are highlighted in yellow, and then look at this mass spectrum here for an example for the entry number 9, is that the Wellington version only has a 4 Dalton, a 2 Dalton shift, but the Cambridge Isotope Laboratory version has a 12 Dalton shift. And so when you're isolating, fragmenting things in the mass spectrometer, when you're doing, when we were using the Wellington standard, we had to really button down our quadruple isolation window, which really killed our transmission. So it hurt our detection limits. By having these larger mass shifts for these compounds allows us to open up the quad to really get much better signal and therefore detection limits.
2 minutes
so we've also,
Dave.
What's that?
2 minutes to go.
Oh yeah, I'll be there. We have a new method on the Exploris 240. We also looked at column chromatography. So the left-hand side was a method that was running at NC State and also at the EPA. You can see the chromatography here is really not chromatography. It's actually very retentive, and it's hard to get the PFAS molecules, the longer chain ones, to come off the column at all. We also looked at the ResTech column. It actually looked pretty good. Then we developed some new column stationary phases, and they actually did much better. So that's what we're moving forward with. The calibration curves, this is linear range is from 5 to 10,000 nanograms per liter with a limit of quantification of about 5 nanograms per liter. In the PFAS world, most of their limits are expressed as 5 nanograms per milliliter, but our detection limits are so much more superior than what's in the literature that we've just moved it to nanograms per liter. Just because if you're reading the literature in this area, you see 10, you're going to think it's exactly the same as what's been done before, but it's actually orders of magnitude better. And so these are our initial conservative limits of detection using these methods, and they're all looking really good. The one that's really tough is Gen X. It's actually detected in its decarboxylated form, but we found that in methanol has a huge background level of contamination of Gen X. So there's an interference there And so we're now switching everything to acetonitrile. Actually, we have, and we've revalidated all these numbers, and GenX most certainly did improve. We do all this with the PFAS world with the help of Mike McCoss and Brenda McLean. Skyline has been around for a while, mostly driven towards proteomics and then moving into small molecules. Now we've adapted it for PFAS, and so we can do all the PFAS quantification and quality controls in Skyline. We also, these are just showing you some examples here of PFDA with the internal standard. We can use the exact mass, we can use fragment ions, we can look at fragment ion abundances here in Skyline, and just make sure that everything's looking really good. And we use NIST SRM materials to as our QC samples. So lastly, we have now validated this entire method in serum samples, which was no small feat because we have to hold this up to the EPA method for 537.1. So we have a full calibration curve, we have a negative control, an 800 nanogram per liter control, and then a standard reference material, which NIST also provides, which is a serum sample. And we repeat this 7 times. The method's really simple for serum. You just crash out your proteins with 3 times the volume of cold methanol with the internal standards of the mixes from CIL, whether it's 1 or 2, and then you do a centrifugation, and then you can make your measurements. When you do this, we get really nice numbers. The way that EPA requests that we report these is minimum reporting limits. So they do change a little bit, and then there's your detection limit. So they get a little bit higher than in the NEAT standards, what you expect. But we did run the NIST SRM 1957, which is a certified standard based on consensus in the field. And we've run that 10 times, and it passed each and every time with our new method. So this was really empowered by the collaboration with CIL and making all these mixes in relatively a short period of time. So I have to thank all the folks there at CIL, including people from that have retired, William Wood and Ron Trollard. I mentioned today the neurotoxins that we're working on, the quantification of PFAS, but also we're working with CIL on an absolute quantification of bile acids, as Andrew talked about earlier. There'll be a kit coming soon, and we've actually just recently re-updated the chemistry and the protocols for doing relative quantification of N-link glycans. And so that's also available from CIL. So again, so thank you for the invite today, and I'd be happy to answer any questions if there's time.
Thanks, Dave. That was an excellent talk. We're a little short on time, but let me see if there are any questions from the floor. It doesn't appear to be, but I'll ask a quick question. I'm interested if you've applied the metabolomics CEMS method from the first part of your talk to search for BMAA and other metabolic markers of ALS in CSF samples.
So your question was, have we done it in CSF?
Have you done it in CSF?
Yes, we have. The CSF samples, though, were done by traditional LCMS methods because it's a technical thing. We got a new mass spectrometer. The old zip chip didn't fit on the front of the new one, so we had to buy a new zip chip, and it's not here yet. So we did it by LCMS, but fundamentally it could be done by the CSF could be done by the zip chip also.
All right, great. Thanks again, Dave.
Sure thing. Thank you.